Hi class, welcome to lab number 10. This lab is going to focus on classical genetics. I want to send out a quick welcome not only to my students, but also any other Bio 107 students that may be using this video to help them with their classical genetics lab. And I know that many of you are using the Campbell text, which refers to chapter 9 of the Campbell text, and some of you are using the OpenStax textbook. So be sure to read the chapter of the textbook that your particular professor is assigning. I also recommend that you watch all of the Amoeba Sisters video that is assigned in your lab prior to watching this video and trying to complete the practice problems in this video. It'll make your life much, much easier, I promise. And I also recommend that you do the genetics interactive activity prior to doing this video. That's the activity where you have the little snurfles. You had the mom snurfle and the daddy snurfle, and they fell in love and had snurfle babies, and you got to construct the Punnett square. And you went over a lot of the vocabulary terms that we're going to be using in this video. So if you haven't had a chance to do that yet, I would pause and go out of this video, do those really quick, and then come back. And I promise this video will go much, much more smoothly for you if you do that. Now, the whole point of this video is to help you get through the Lab 10 lab quiz. So if you have your lab manual handy, I would recommend that if you don't, pause the video really quick and go grab your lab manual and turn it to page 117. The idea here is that I'm going to introduce and clarify some of the lab questions that are found in the manual, and those are all the questions that you'll have in the quiz. So if you pay attention throughout this video and write down the problems as you're doing them, once you take the quiz, you should be able to finish the quiz in about 5 minutes. It should be really easy for you. Okay, so let's go ahead and review some of the terminology that we'll be using prior to actually doing some of the Punnett square problems. So of course, first and foremost, we want to talk about genes. Remember that genes are a unit of inheritance for a character. And students often get the terms character and trait conflated. They get them confused. They switch one for another. Remember that character is the category of which traits can be in. So if the character is eye color, for example, a trait would be blue eyes, green eyes, brown eyes, and so on. Now, all of our genes are found in our chromosomes, and they're found in a particular spot in our chromosome. So all of us, for example, have the similar genes in the same location or loci. That's plural for locus. And locus, if you see the word right here, locus is really just a fancy science term for location. So locus basically means location. It's in the same spot. So a gene is found in the same spot on all human chromosomes. All of us have the same gene. Well, we don't have a gene for flower color. This is a gene in a chromosome in a plant. But for example, the genes that code for our eye color are all found in the same location on the same chromosome in all humans. So when we have chromosomes, we can refer to those chromosomes as autosomal. And autosomal, we have 22 pairs of these autosomal chromosomes, and each chromosome has a copy. We get 2 of them, one from mom and one from dad. And on these chromosomes are found, of course, our genes. And the gene that's found in the same place on homologous chromosomes, right, one from mom and one from dad, are called homologous genes, right? So we have our homologous chromosomes, and then we have these 2 genes, 2 variations of a gene, I guess I should say, and those variations are called alleles. So this particular gene has 2 different alleles, an allele for purple flowers and an allele for white flowers. Now, if these 2 alleles were the same, if they coded for the same trait, we would call those alleles homozygous. Remember that homo means same. And if they're different, we would call them heterozygous. So these 2 are different. We have one gene that codes for a variation of purple, and we have another gene that codes for a variation of white. So these genes would be considered, or this set of genes, these alleles would be considered heterozygous because they're not coding for the same trait. Now, to further further clarify how genes play a role in trait inheritance, we want to talk about dominance and recessive qualities. Now, dominant alleles Oh, I meant to look this up. I forget. I want to say purple is dominant. It doesn't really matter, but I believe that the purple color, I should know this, the purple color should be dominant in flower colors if we're talking about pea plants, for example. Of course, the famous Mendel experiment. And if a gene is considered, or I should say an allele is considered dominant, we're going to capitalize the symbol for that allele, and it's always expressed. So if a dominant allele is present in an organism, that allele will be expressed. So for example, here, if purple is dominant, then the flower color will be purple, even though we have another allele that is coding for white flowers instead of purple. On the other hand, we have alleles that code for recessive traits. So these recessive alleles, we're always going to symbolize them using a lowercase letter. And the only time a recessive allele is actually expressed is if both copies of this particular gene are recessive. So in a dominant, we can have basically three different situations. We can have a homozygous situation in which both alleles are the dominant allele. We would consider that homozygous dominant. So for example, if our symbol for flower color is R, capital R would be considered homozygous dominant, and the dominant trait is expressed. On the other hand, we can have an organism that is heterozygous, like this one. This organism here is heterozygous. It has one dominant allele and one recessive allele. We would consider this capital R, lowercase r. It's heterozygous, but remember that the dominant trait would still be expressed, because if that dominant allele is present, that's the trait that will be expressed. It's only when we have a homozygous recessive situation where we have the recessive trait being expressed. And here we would have two lowercase r's to symbolize that both of those alleles are the recessive allele for whatever trait it is that we're looking at. Whoa, my computer almost fell. Okay, so I've talked a lot about where genes are, and they're located on chromosomes, one chromosome from mom and one chromosome from dad. So let's look a little bit further into these chromosomes and discuss how chromosomes actually work. Remember that humans, we have 23 pairs of homologous chromosomes. In other words, we have 46 total chromosomes, but of those 46, they're found in a set. We have 23 pairs. And it's actually not technically true. We have 22 pairs of autosomal chromosomes. So of those 23 pairs, 22 of them are considered autosomal chromosomes, which basically, what does that mean? They're not chromosomes that relate to sex inheritance. So that last pair of chromosomes are considered our sex chromosomes. And they're not, some people would argue that they're not always necessarily a pair. So let's look at how this works. In females, most females anyway, have an inheritance pattern that we get an X from mom and an X from dad. So these X chromosomes are true homologs. They look exactly the same. They have, I shouldn't say exactly the same. They might have differences in their sequences, but the length is the same. The gene locations are the same. In other words, they have similar alleles or similar genes in the same location on each chromosome. So again, the mom gives us an X and the dad also gives us an X, and that would lead to a female offspring. On the other hand, mom can give us an X. Mom will always give us an X. But dad might give that offspring a Y chromosome. And X and Y chromosomes are not true homologs, because as you can see, the X chromosome is much bigger than the Y chromosome. And therefore, genes that are on the X chromosome have no counterpart it's missing, right? There's nothing there on the Y chromosome. So in the case of male offspring in humans, I'm not sure how many other animals use this kind of sex-linked inheritance pattern. You can see that in the case of male offspring, they have a unique set of sex chromosomes in that they have a Y chromosome from, an X chromosome from mom and a Y chromosome from dad. And this shortened Y chromosome actually has a lot of ramifications for inheritance, as we'll see later in the lecture. So the whole point of classical genetics, this lab and chapter 9 for us, is talking about being able to predict the traits that offspring will have. Remember that the big advantage of sexual reproduction is that we can introduce variation in our offspring. And this happens in three main ways. The first two happen in meiosis, crossing over, which happens in prophase 1, right? When tetrad formation occurs, we have crossing over occurring between the homologous chromosomes. And we can also have in when the when the tetrads line up in metaphase 1, we can have independent assortment, which basically means that when the chromosomes, those tetrads line up, they're doing so independently of one another. So we're essentially shuffling a deck of cards every time those chromosome pairs line up along the metaphase plate in meiosis. And both of those have huge ramifications for genetic variation in that particular gamete, in that sex cell, that egg, the ovum or sperm cell. And then a third way of introducing genetic variation, of course, is at the fertilization event itself. Remember that there's millions and millions of sperm in any fertilization event, and during the course of, say, a female, at least in humans, right, a female's reproductive efforts, it could be potentially dozens of different eggs during that span of time. And so the combination, that unique combination of sperm and egg that ultimately successfully form that zygote, introduces variation as well. So we have all of these mechanisms that are going to produce lots and lots of variation in our offspring. And the question is, can we actually predict, based on our genes, what traits our offspring will have? And it turns out that we can. So again, let's go ahead and grab your lab manual and turn to page 118 of your lab manual. And I want you to pause the video at this point and read, starting on page 118, I want you to read crosses involving a single gene in the monohybrid cross, and then quickly read the directions that are on page 119, the steps to take in order to work a genetic problem. So pause the video, read through that roughly page and a half of instructions, and then let's get started on our first problem here. Okay, so this problem is modeling the exercise 1 that starts on page 119, and this is all about monohybrid cross with a single dominant gene. So we're assuming that we have a single gene that has only 2 allele possibilities, one allele being dominant to the other. So we can observe here we have 2 different bottles, and in one bottle, I promise they're not I promise they're not this big in real life. I just made them big so that you can see their wings a little bit easier. But in one bottle, in bottle A, we have a true breeding fly that has vestigial wings. So they're tiny wings that actually aren't functional. And in bottle B, we're going to have true breeding flies that have a normal wing structure. So we have bottle A and bottle B. And remember, this is a very important hint. It says true breeding. If you recall from the Mendelian portion of our lecture, true breeding implies that these particular organisms are homozygous for that particular trait. So, from this, we need to go a little bit further. So bottle A and bottle B, the flies from each bottle were allowed to mate, and bottle C shows their offspring. So I put lots of their offspring here in bottle C, and I want you to observe these flies and ask yourself which is the dominant characteristic and which is the recessive characteristic based on this outcome. So again, here are your parents. We have a fly that has vestigial wings, true breeding, and a fly that has normal wings, also true breeding. These 2 flies were crossed, and their offspring are shown here. So ask yourself, go ahead and pause the video, ask yourself which is the dominant and which is the recessive trait, and write those in on questions 1A and 1B on page 119. And let's go ahead and continue on and let's assume just for, you can really choose any letter you want, but let's assume just so that we can all work together on this, that the symbol for normal wing is capital N. That should maybe give you a hint as to which one is dominant, is capital N, and the symbol for the vestigial wing is lowercase n. So the parents' genotypes in this particular case, right, in the parent number 1 in bottle A would have, because it's true breeding, it's homozygous for lowercase n, lowercase n. And the parent number 2 in bottle B would be homozygous for the dominant trait, capital N, capital N, because remember, they're both true breeding. So let's assume that parent number 1 is homozygous recessive and parent number 2 is going to be homozygous dominant. All right, so let's try our first Punnett square on page 120 of the lab manual. So you can read the question here. This is question 2. It starts at the bottom of page 119 and then goes up to the top of page 120. And I actually set up the Punnett square for you here. We have parent number 1 that is homozygous dominant for wing shape, and parent number 2 is going to be homozygous recessive. So go ahead and complete this Punnett square on your own. Again, this is at the top of page 120, and use this Punnett square to answer questions 2A and 2B in your lab manual. All right, let's move on to question number 3. So we're really doing the same thing. We're crossing one type of trait, monohybrid. So a hybrid, remember, means essentially a crossing of 2 species, and we're only looking at one trait. So it's called a monohybrid cross. And here we're just going to change the parent's genotype up slightly. Parent number 1 is going to be homozygous recessive, so they have lowercase n and lowercase n as their 2 allele possibilities. And parent number 2 is going to be heterozygous for this same character. Remember, we're talking about wing shape. So again, this is in the middle of page 120. I set up the Punnett square for you here. Complete the Punnett square and use that completed Punnett square to answer questions 3A and 3B. Okay, so now before we move on to more complicated Punnett square questions, let's do one more Punnett square problem based on this idea of complete dominance. We have this one allele that is we have a gene that only has 2 allele possibilities, and one of those possibilities is always dominant to the other. So let's skip ahead to page 126. And let's try practice problem number 1 on page 126. I want you to answer again. So the question we have, this is a real issue. So for some parents, when they have a baby, that baby can be it's called PKU. It's a phenylketonuria. It's an inability to make, I believe, a certain type of amino acid. And as long as doctors are aware of this trait, they can fix it, no problem. But if they're unaware, the baby can suffer some pretty serious consequences. So this is one of the screening events that take place in infants. And so one of the things that they're going to do is first screen the parents to see what their genetic markers are like. So if we have the 2 heterozygous parents, we have parent number 1 that's heterozygous, parent number 2 that's also heterozygous. Let's assume that the symbol for PKU is capital P and lowercase p. Remember, they're both heterozygous. And the offspring would have to be homozygous recessive in order to actually express this trait, because this is a recessive, an inherited recessive disorder. So again, complete the Punnett square that you find that I set up for you here, and use this completed Punnett square to answer questions 1A, B, and C. All right. So let's move on to more types of genetic problems. And of course, it's never that easy. If only we inherited all of our traits as a simple complete dominance type of problem, genetics would be very, very straightforward. In fact, that's not the trait. There's lots and lots of complicating variations on this idea of inherited traits. And we're going to talk about a few of them here in this video and do some practice problems that show how they work. And so the ones that we're going to talk about are these concepts of incomplete dominance, codominance, traits that are sex-linked or sex-linked inheritance. And then we're also going to talk briefly about the idea that you can actually have multiple alleles. There's more than 2 allele combinations that are two possible alleles that can be found on any given gene. All right, so let's talk about incomplete dominance first. Incomplete dominance can be found on page 124 of your lab manual. And incomplete dominance, I think this is part of what stymied a lot of early experimenters in genetics. So this is kind of similar to the blending hypothesis that many scientists believed in up until the 19th century with Mendel's advances. And that is the idea that if you combine 2 factors, you're going to get some factor that's kind of in the middle. So it's almost like the paint if you combine blue paint and red paint, you're going to end up with purple paint. And that's really what happens here in this idea of incomplete dominance. There's no true dominance from either allele. So there's a type of flower called a snapdragon. They're actually pretty common out here. They're really pretty. They're, at least in the Midwest, they're annuals. I'm not sure if they're annuals out here or not. But there are variations on the color of snapdragons where you can have a red flower and a white flower. And if you were to cross these 2 true breeding colors, you would end up actually with a pink flower. So you're truly blending the phenotypes of these 2 genotypes, right? The genotype for this one is, the color is red, so it's homozygous red for this flower. Whereas this flower is homozygous white. So the only possible combination that their offspring could have would be heterozygous for red and white. And so they end up neither red or white, but some combination in between those 2. Of course, in this case, it's pink. So we tend to symbolize incomplete dominance alleles with a lowercase letter since neither allele is dominant. Another example of a variation on inherited traits is codominance. And this is if we have 2 different alleles and both of those alleles are present, then they're both expressed, but the key part of this is that they're expressed independently of one another. And so many students get this confused with incomplete dominance. Let's look at these 2 side by side. Here's our picture from the previous slide, our model of what incomplete dominance looks like. But now let's look at an example of codominance. Here we can see that we have a, I'm not sure which one is the mom or the dad. Oh, this one's the mom. It has udders. Okay, so this is the mom. The cow is homozygous for white fur. So you can see, I don't know if you can see this too well, but it's the symbol is RW. Whereas the bull is homozygous for red fur. So it's R, and R. So when these 2 are crossed, their offspring there's only one possible combination that their offspring can have, because they can only contribute, each of them can only contribute in the case of the cow, the RW allele, and the dad can only contribute the RR allele. So their offspring will be RRRW. And what this results in is a type of fur pattern called roan. And as you can see, roan has elements of white fur and red fur. This is codominance. So I want you to pause the video here and ask yourself if you can describe what the difference is between these 2 types of inheritance. Here we have incomplete dominance, and here we have codominance. Can you say in words, can you actually describe in words what's happening here? Okay, I'm going to assume you did it. I know you did it. I have faith in you. And I'll help you out a little bit here. in incomplete dominance, there is a third trait that is expressed. Remember, traits is the physical manifestation of the phenotype. So the phenotype here is red, the phenotype here is white, and when we have incomplete dominance, we're actually going to get a third phenotype, a third trait. And in this case, that trait is pink. Now, on the other hand, in codominance, what's going to happen is we're not necessarily getting so much as a third, we're not getting a third trait. What we're getting are both of these traits being expressed at the same time. So this cow has both white fur and red or brown fur, whatever color this fur is here. So very important difference between codominance and incomplete dominance. One, in incomplete dominance, we have a third trait being expressed. So let's actually try a codominance problem here so we can see what this looks like. And we'll use our roan fur example. So let's go ahead and turn to page 127. And this is question 9. And again, at least if you're in my section, chances are you want other 107 students as well. This is going to be your quiz. So you want to pause the video while I'm explaining this to you so that you can have the answer ready when you take the quiz. So let's look at question 9. Go ahead and pause the video, read through that problem. And the problem ultimately is asking you to predict the phenotype and the genotype for each of these offspring, or what are the possibilities, right? What percent will have this color fur and so on. So we have a white cow. Remember that white, I'm going to change these symbols really quick for you here. Hold on. Actually, I'm not going to change them because this is how they are in the problem. So this will work for us. So white cow has a lowercase r. So this particular white cow is homozygous. Let's see here. But with white cows, and we know it's homozygous because it's white, right? The only way for it to be white is if it's homozygous for each of the allele. And we have a roan bull. So what does the genotype of the roan bull have to be in order for it to have a phenotype of roan fur? Remember that in a codominance problem, this bull has to be capital R and a lowercase r. And I think I am going to put that in for you. Let me fix this really quick. There we go. Let me go full screen. Oh, no. Why did... Darn autocorrect. Autocorrect is not fair to scientists because we always are doing weird things with capitalization. Okay, great. All right, so now we have our roan bull, who of course has to be heterozygous because it's roan, right? The only combination that can lead to a roan bull or a cow, or a roan cattle in general, is capital R, lowercase r. So based on this Punnett square and the idea of codominance, I want you to complete this Punnett square for the roan bull white cow cross and predict the genotype of of their 4 offspring. And once you know the genotype, of course, you can predict the phenotype of fur color as well. All right, let's move on to our next concept of, again, complicating factors that have to do with dominance in inherited traits, and that's the idea of multiple alleles. Remember that a human can only have 2 versions of a gene. However, there can be many versions of that gene within a population. So even though you get 2, those two different spots on your chromosomes can have any number of different gene possibilities that you got either from your mom or from your dad, of course. And a perfect example of this is blood typing. So humans have 3 different allele possibilities. We can have a gene that is IA, a gene that is IB, and then finally a gene that is lowercase I. So remember, we can only have 2 of them. We can have a combination of any 2 of these 3 possibilities. So let's look at this in a little bit more detail. Blood typing is a really good It's a fun genetics problem. It has a lot to do with physiology and anatomy, and we even talk about this a little bit in microbiology because it ultimately has ramifications on immunology as well. This is why blood typing, for example, is so important, because if you receive a blood transfusion of a blood type that's not your own, your body will have an immune reaction to it, and in some cases that immune reaction can be quite serious. So what is that immune reaction based on? Well, your body recognizes very specific markers on the outside of your red blood cells. And so the allele that has IA codes for a marker on the outside of your red blood cells that looks, let's just say, like this. And then if you have the IB allele, it's going to code for a marker that looks like this. And then the lowercase, finally, the lowercase I leads to actually nothing. So there is no marker if you have the I allele on the outside of your red blood cell. So blood typing shows examples of all three of these concepts: dominance, codominance, and multiple alleles. And let's look at how this works. So remember that we can have 1, 2, 3, 4, 5, 6. So there's 6 different possible combinations that a human can have in terms of their allele combinations. If a human inherits IA from mom and IA from dad, they will end up with type A blood. In other words, this will be the only marker that they show. Now, if they were to inherit IA from one parent and the I allele from another parent, this I allele, because it's lowercase, remember, is recessive. So they're still going to be type A blood. Now, it's very similar for B. If I get IB from one parent and IB from another parent, I'll have type B blood. If I get IB from one parent with the recessive lowercase I allele from another parent, I'll have type B blood also. So far, straightforward. We can either be homozygous for either blood type A or B, or we can be heterozygous with this lowercase I allele. But let's look at what happens if we have either of these other two combinations. If I get IA from mom and IB from dad, I will have a blood type that has both of these alleles expressed. In other words, I'll have red blood cells that have both these yellow, or what color is it? Pink circles and these blue square diamond things on the outside of my red blood cells. So I will have type AB blood. This is an example of what? Is this dominance, codominance, multiple alleles, incomplete dominance? What is this? This particular one, IA and IB. This, of course, I'm hoping you got this right. This is a perfect example of codominance, right? If I inherit each of these alleles, both of them will end up being expressed. It's kind of like the roan cattle. If I have one from mom and one from dad, I'm going to end up expressing both of them. So only when we're homozygous recessive for lowercase I, in other words, we get the lowercase I allele from both parents, will we have nothing expressed on the, none of these markers, I should say, expressed on the outside of your red blood cells, and you will be type O blood. So we have multiple alleles. We have 3 different allele combinations. In some cases, we can see dominance occurring, for example, here and here. In other cases, we can see an example of codominance right here. So let's try a problem with blood typing. This is a fun one. This is like a logic puzzle. Let's see here. It's on page 127. It's question 7. So I went ahead and set up this problem for you. Go ahead and read through this, pause the video, read through this question, and then I'll kind of explain my setup right here. Okay, so if you read through the problems, what we're ultimately asking is, did the right parent couple get the right baby? And so we have mom and dad Jones. They're both type A blood, and they have a baby Jane, that ends up with type B blood. On the other hand, we have mom and dad Smith. Mom has type B and dad has type A, and they have a baby Shirley that ends up being a type O blood. So the question you have to ask yourself is, did the right baby go home with the right set of parents? In other words, are the babies switched? And in order to understand this, or in order to answer this problem, you have to ask yourself, we know what the phenotype of the parents are. We know that the blood types of each of the parents. What you have to ask yourself is, what are the genotypes of these parents? So what is the genotype of mom, possible genotypes, right? Mom Jones has type A, so she could be 2 different genotypes, as could dad. Now, Mom Smith is type B, so she could have either of these 2 genotypes, whereas Dad Smith is type A, so he could have either of these 2 genotypes. So you have to ask yourself, are any combination of those genotypes, could they lead to the blood type of these babies? And if they can't, then we know that these babies had to be switched. It's also possible that you're not able to tell. So before genetic testing was common for babies to determine either paternity or both parents, for, like in the case of a switched baby, the only way that we were able to tell before DNA fingerprinting kind of became a more common and usable technique was through blood typing. And so sometimes blood typing doesn't work. So the question that we're asking here is, did it work in this particular case? So answer this question, pause the video, answer this question, and then let's try another blood type question. So let's look at question 8 on page 127. Right below question 7. Here we have a man and a woman. The man is type IA and then lowercase I. So he's what blood type would this be? Blood type A. All right, he's blood type A. The woman is blood type AB because she has IA allele and the IB allele. So there's an example of co-dominance here. So we have the Punnett square set up with the man and the woman, and the question is asking, what are the possible blood types so that their children can have? So here you're going to determine the genotypes of 4 potential offspring or any combination of their offspring. And from their genotypes, you can predict what the phenotypes of their offspring may be. All right, let's keep on going. Another example of codominance, which is found on page 124 of your lab manual, is the disease or the condition of sickle. I shouldn't call it a disease because it's not communicable. I guess I'd call it a condition of sickle cell anemia. And sickle cell anemia has 2 alleles, HbA, which codes for normal hemoglobin, and HbS, which codes for abnormal hemoglobin. And so we have these 2 allele combinations. And of course, if we're heterozygous dominant, should I call it? I can't say dominant because there is no dominance, right? They're co-dominant. So if we're heterozygous for A, for homozygous for A, the offspring will have normal hemoglobin. The person that has HbA will have normal hemoglobin. On the other hand, the person that has both alleles as HbS will have the condition of sickle cell anemia. Now, of course, we can have another combination, which is heterozygous for this trait, HbA and HbS. And this person, even though they don't have sickle cell anemia, they are a carrier. And no, sorry. So what this means is that roughly half of their red blood cells that are expressed have normal hemoglobin, and the other half will have this characteristic that leads to the sickle cell pattern in their red blood cells. So again, this isn't complete dominance. This is an idea of co-dominance because both of these traits are being expressed in a heterozygous individual. So if we do a problem that involves sickle cell anemia, question 12 on page 127, the question is asking if we have one parent is going to be heterozygous for sickle cell. So this parent is a carrier, right? It's HbA, HbS. So this parent has the sickle cell trait, and the other parent has normal hemoglobin. So they have HbA, HbA. So the question is, what is the probability of any of their children ending up with sickle cell anemia? And what is the probability that any of their children will end up being a carrier? In other words, they'll have the sickle cell trait. So go ahead and complete the Punnett square and predict the probability for those offspring. Okay, so let's look at our last concept here in terms of inheritance, and that is the concept of sex-linked traits. Remember that in my introduction to this video, we talked about how females can only ever contribute the X chromosome as the sex chromosome, as the sex determinant chromosome, but males can ultimately determine the sex of the offspring by either contributing an X, which would lead to a female offspring, or contributing a Y chromosome, which tends to lead to a male offspring. Now remember also that the Y chromosome is much smaller than the X chromosome, so the Y chromosome doesn't have as many genes as the X chromosome. So here we can see this is a duplication of the diagram on page 125, and you can see that the sperm can either contribute X or Y, whereas the egg will always contribute X. So what ramifications does this have in terms of traits that are, how should I say this? Traits that are linked to alleles found on your sex chromosomes. Well, what this does is it leads to a differential inheritance pattern. So for females, we always have 2 copies of every gene because we have all our autosomal genes are paired, and our sex genes, our sex chromosomes are also paired because we have 2 copies of the X chromosome. Whereas males have only 1 copy of those genes that are found on the X chromosome because the Y gene, or the Y chromosome doesn't have them. So this leads to a couple of known problems in males. The 2 that we talk about in this lab manual are color blindness and hemophilia. So the one that we're going to actually do a problem on is color vision and color blindness. So remember that the definition of a recessive allele or a recessive gene is that the recessive trait is only expressed if both alleles have that recessive gene found on them. So since only the X chromosome carries that gene for color vision and the Y gene doesn't carry it, then the females have to be homozygous. In other words, both of those X chromosomes have to carry the recessive allele for a female to be colorblind. Whereas for males, only the X chromosome in the mother has to carry that recessive gene because there is no gene on the Y chromosome. In other words, there is no paired gene that can be dominant in a heterozygous. In other words, there's no heterozygous-ness for males. There's no allele for that particular color or for that particular trait, in the Y chromosome. So let's look at an example here with color vision. Remember, so X capital C would be the normal color vision allele, whereas X lowercase c would be the color blindness allele. So this allele leads to color blindness. So remember that females have to be homozygous for the recessive trait. So in other words, the only way that a female can be colorblind is if they're homozygous recessive for this trait. Whereas a male can only if it remember that how am I saying this? The male only needs 1 copy of that recessive gene in order to be colorblind. So this leads to more males having the colorblindness trait than females. So you can see other combinations here. So for example, a heterozygous female can have a capital, X capital C, X lowercase c. And in this case, even though she's not colorblind, she is carrying that colorblind allele that she could potentially pass on to her offspring. So let's try an example of color vision. This is question 18 on page 128. I want you to answer all 3 kind of sub-questions that are related to this. And in order to understand this question 18, we're going to take a man with normal color vision. So he got an X from his mother, He has the X capital C, so normal color vision from his mother. And of course, he inherited nothing from his father because the Y chromosome doesn't carry that particular gene. Whereas the mother is a carrier, I think. Let me look. No, she's colorblind. So she's X lowercase c, X lowercase c, So she's a colorblind female. So she has this genotype here. Whereas the male is this one right here. So do your Punnett square cross and determine what proportion of their children are expected to be colorblind, what proportion of their daughters will be colorblind, and what proportion of their sons will be colorblind. And I think that's the last. Yeah, you're done. Congratulations. You now know everything there is to know about genetics. Just kidding. You only know a small part. So this is good. This hopefully gave you a nice introduction to how probability and predictions work in very, very simple genetics problems. Of course, as you can imagine, genetics problems get way more complicated than what we just did. And this just kind of gives you an introduction to the field. It's actually a really cool field. If you like math and you kind of understand statistics and probabilities and you can make predictions based on data comfortably here, you might want to think about looking at the field of genetics, or maybe looking into genetics counseling as a career option for you. Okay, so again, make sure that you're doing all of the practice problems that I went over in this video, because they're your problems for your quiz. Good luck.